Wow, that was such a nice and great, thorough introduction to what I want to talk about. Thank you so much for having me here. It's such a pleasure to be back at University of Maryland today. You know, we get so busy in our lives and we don't go back and reflect on where we came from. So it's really nice to be here, and I've enjoyed the day very much. This Bioscience Day has been great. So my talk is a little different from the previous talk. I do modeling and I do neuroinformatics, but even if you're not that interested in modeling, I think there will be something for you today. So all of us who work in science gather data, we do data analysis, and anytime you do any kind of computational work at all, whether it's modeling or data analysis, I think some of the things that I bring up will be things that will kind of resonate with you, and a lot of the tools that I'm going to talk about today, the resources, there are parallels that are out there for the neuroimaging community, for the neurophysiology community, and so if you're interested in that, I can give you information after my talk about other areas, but I'm going to talk more about computational neuroscience today. Okay, so I'm going to start with, the model, right? Everybody knows about the famous model from Hodgkin and Huxley. And so in 1952, this great, amazing article came out. And I think it really still holds up today. If you go and look at this article, one of the things that really stands out is that it's so thorough. They have these appendices where they talk about every method they used for every computational thing they did. What also, of course, is amazing to those of us now who have these, computational resources at our fingertips is that they did all this amazing work with slide rules, right? And so, but the point I wanted to make today is that in their, in this original sort of model, it built on the work of many people before them, but also it was very fundamental to making leaps in our understanding of neurophysiology. This simple model, today we think of it as quite simple because it has 4 differential equations, 4 variables, a couple dozen parameters, constant values that have to be measured based on experimental data. This is what they did so well, and it is explained very well in their paper. They did all these amazing experiments in squid, giant axons, and the data are available in the paper. You can see the fittings that they did, and everything is laid out perfectly in the paper. So today, if you want to go and implement the Hodgkin-Huxley model, it's quite simple to look at their paper, get the equations, get the parameters, understand everything about what they did. Everything is there. And of course, 10 years later, they received the Nobel Prize for this seminal work. Contrast that to a model from today. Not that long ago, an entire issue of the journal Cell was devoted to a model coming out of Henry Markram's lab, the Blue Brain Project. Some of you might have heard of this modeling that they're doing. And so one example, they have many models for one cortical column, multiple cortical columns, 10, hundreds of cortical columns. But in this example, it's about 0.3 square millimeters of the rat somatosensory cortex, 31,000 cells, 55 different morphologies, so that's the shapes of the cells that they used. And actually for their morphologies, they have variability in their morphologies, so even though it's only 55 sort of morphology types, they have many more different morphologies in their model. They have 207 combinations of morphology with electrophysiology, so different morphology types. They've done experiments and they see that the neurophysiology is different, and so they have basically 207 different cell types in their model and about 37 million synapses. Okay, well, I don't think we can all go and reproduce that. Number one, we don't have the computational resources, but even if we just wanted to reproduce one of the cells in their model, we would have a hard time. It's tens of thousands of equations are involved. We would want to know what are all those things in the equations, what do the variables represent, what are the parameters, what are the units for the parameters. And even though they did a pretty good job in this journal article, we can't get everything we need from the journal article itself. Here's the reference in case anyone is interested. Okay, so this points out, this is at the extreme end of this idea of maybe there's some problems with reproducibility in computational neuroscience as well as experimental neuroscience. And so, you go to the Society for Neuroscience meeting, any meeting these days in science, and you'll hear a lot about reproducibility. And there are a lot of efforts out there right now to make tools for people to use in their lab, to make databases for people to share things. And so I think we're making really positive strides. I've seen a huge change over the last decade in science in terms of moving towards open science, moving towards open publishing, and a lot of other things. So I think it's very, very positive, a lot of very positive things going on. But I wanted to point out this paper that came out quite some time ago that rang true for me when I was a graduate student and even beyond, because as models become more and more complex, it really is difficult to reproduce what somebody describes that they've done with their computational work. All right, so what are some of the things we might want to think about if we want to promote reproducibility? And so if we agree that I think we can all agree fundamentally that reproducibility is important. We might disagree on how we need to do it, but it has to be important because reproducibility really is the cornerstone of science. We need to be able to build on the work of others. In computational science, I think one of the things that we appreciate is that we can take the computational work of others, we can improve it, we can reuse it, we can improve it, we can build on science and make new discoveries by using what other people have done before us. And so there are three key things, I think, for reproducibility in computational science and maybe in other sciences as well. So one is accessibility. So as I showed you with the blue brain model, one thing that would be really helpful is if we didn't just have this publication, but we also had code and implementations of models so we can build on that. That's sort of what I mean by accessibility. Can we access the code? Can we access the software that people are using? In data analysis, it's the same thing. Are the software products that people use open source? Can I get them? Can I reuse them? Are they accessible? Another issue is portability. So for a model, one problem we sometimes have is somebody implemented that model in, say, MATLAB. I don't want to use MATLAB. I want to use some other existing simulation software like Neuron or Genesis, or maybe I just want to use Python. Whatever it is, I have this portability problem. I can't just take code from someone and necessarily rerun it and do the things that I want to do. So we'd like to improve portability. And then another issue is transparency, and I kind of alluded to this before. So just giving me the code for the Blue Brain project may or may not be that helpful, because if they have millions and millions of lines of code, that's not really what I want. What I really want to know is, what are the equations, what are the exact variables, what are the exact parameters, what are their units, so that I can do whatever I need to do with that. And that's kind of transparency. I need to know, exactly what those things are that are represented in the code. Another issue of transparency is basically this idea that comes across like when you write a methods section for your article, right? So when I write a methods section for some experiment that I did, I have to be sure that somebody could do exactly what I did before, and that's really what transparency is about. I don't want to hide some step that I did in my science. I want everyone to be able to redo my science. Okay, so that's another aspect of transparency. So the accessibility part, is basically a lot of people are trying to address. And so, for instance, in computational neuroscience, at the level of sort of biophysically realistic models, spiking network models, not necessarily cognitive level models, although some of them are here. One of the resources that people often turn to is ModelDB. And so ModelDB has many different models. I don't think it says on this slide, but they have well over 1,000 curated models. So these are models that have been published. You can go to ModelDB. There are a lot of different ways to search for models. You can find them. You can get the original code for those models. All right, so it's a place where you get code, right? So some of the models are available in Neuron or Genesis or Nest. Some are available in Python or C or MATLAB. But it's a starting point, right? It's great that we have all these published models that we can build on. And throughout my talk, you'll notice that when I talk about these online resources, these software, you'll see something like this. So I'll try to give the URL, but I also give the RRID. And if you guys don't know about RRID, this is sort of a registry of resources. So for instance, if you publish a model and you used Neuron to run the model, then you should give the RRID for Neuron for your model. Or if you got your model from ModelDB, you should give the RRID for ModelDB. Right? So it's a way of kind of keeping track of resources. And if you go to SciCrunch and you put in the number for the model, the ID for the resource, you'll get a lot of information about that resource, right? So it's a good thing to have. Okay, so ModelDB is great for accessibility. In systems biology, you can go to sites like the biomodels database, it's much broader than just neuron models or neuroscience models. It has they have a lot of other stuff there. And there are similar examples in other fields. Okay, so I want to talk a little bit about these ideas of portability and transparency. There's a whole tool chain out there of things that are available to people, and I want to let you know about some of these things. If you're interested in modeling, this might be a good starting place. If modeling's not your thing, you'll see some parallels in ideas about data analysis, tool chains in neuroinformatics for neuroimaging and things like that. Okay, so my very nice introduction that was given mentioned NeuroML. So this is a project that I work on, and I'm not going to give a whole lot of details about NeuroML. If you're here to hear all about NeuroML, I can point you to some other online talks that you can watch that'll give all the details, but I don't think they're that interesting for the whole audience. But I am going to try to tell you why NeuroML is cool and what we can do with it. Okay, so an example. Here's a pretty simple model that comes from a paper by Brett and Gerstner. And what you see up here are two differential equations. We have two variables, about 11 parameters, 11 fixed constants. When you run a simulation, they're fixed. And the point that I wanted to make with this slide is when you choose a set of those constants, you choose a set of parameters, you can get very different types of behavior out of this model. So you can replicate behavior that you would see in a neurophysiology experiment when you're looking at the membrane potential, in a bunch of different cell types by changing these parameters. All right, so it's kind of a family of models where when you choose a set of parameters, it's one specific model, it's an instantiation of that model that gives you a specific kind of behavior. So this is a simple model. We can explain it in a paper. We don't really need NeuroML, but I wanted to give you an example where it was clear to you what's going on. So here is how you might describe that model in NeuroML. So NeuroML has a concept of this adaptive exponential integrate and fire model. And there are many, many types of models that are typically used in computational neuroscience where we have this concept in NeuroML. And so NeuroML is a markup language. Many of you might be familiar with markup languages. Maybe you've seen HTML before, which is how you describe, the information that's on a website. And so the thing about markup languages is you have a way of putting the data into the document, and then you have these tags that describe the data. So you have the data, but you also have metadata about the data. And it's the metadata that, that you really, really want. Those are the things that are important. Semantic information about, for instance, in NeuroML, what do the variables represent? Semantic information about what the parameters represent, what are the units, and things like that. We can put all that information into the NeuroML file. But so for an example here, here's a particular example of one of these models where it's one particular parameter set. And here's how it's described in NeuroML. So you have a tag saying what kind of model it is, and then you have the parameters and their values. All right? Different parameter values, a very concise description in NeuroML. That's all it is, right? Okay, underneath NeuroML, there's something that's much more verbose that describes all the stuff underneath, that kind of model. So it says what the variables are, what the equations are. So here are the equations. All that other information is included in this machine-readable thing that's underneath those NeuroML components. All right. So that's probably way more detail than you need, but it's pretty much all the detail that you need. Okay. So the idea is you have a way of describing very complex things that is designed around neuroscience objects. So it's kind of conceptually easy for someone who's thinking about neuroscience. And then you only have to deal with those sort of high-level objects if you want to define a model. The important thing about it being a markup language is that you can create these mappings between the description and all kinds of other things. And so as an example, you can have a mapping to a very nice human-readable output that like lists your parameters and lists what they represent or something like that. So for instance, we have tools where you can take your NeuroML model and you can output a table that you can put directly into your publication that gives, for instance, all the information about the network structure, that gives all the information about the parameters. You can put that straight into your publications. Oh, and then the other critical thing here is that you can have mappings to simulators and programming languages. So your model is described in NeuroML. You can basically just run a quick script, and you can get that model in Neuron code, Python, C, MATLAB, Genesis, Nest, all these different things. So then you choose what kind of simulator do I want to use to run this, and it's there for you. All right, and then I'll just say that there are all kinds of tools for interacting with NeuroML files, for generating these simulator languages. All this stuff is on our website. You're, of course, welcome to contact me if you have more, more questions or want details. We're always happy to answer questions. More importantly, maybe, there are a large number of tools out there that people use that have some level of support for NeuroML. So, for instance, I'll talk about NeuroMorpho in a minute. So this is a database where you can obtain morphology information about cells, about reconstructed cells, in NeuroML format. The OpenWorm project, which some of you might be familiar with, where they're trying to simulate the entire C. elegans and its behavior. All of their neuron models and connectivity is built on NeuroML. So there's just a lot of examples of things like that. This is only a subset here, but you can find all the information on our website about the tools that support NeuroML and how you can use them and people to contact to learn more about it. All right, so here's an example. One example of one of these tools is NeuroMorpho. So NeuroMorpho, right now, I can't even read that number. 700 and something thousand reconstructed neurons that they have. It's really exciting because I've been following this project since they first started putting neurons on there where there were only hundreds. Now pretty much any cell type that you want reconstructions, people know, wow, it's really cool if I did all this work to reconstruct this neuron, I'm going to put it on NeuroMorpho, and we can all benefit from that. We can go to this site and download these morphologies and use them in our models or use them to do morphological analysis. I mean, all kinds of, cool things that we can do. They have tools there, but the point I wanted to make is one of the tools they have is this neuron viewer, and when you open up the viewer, you can then click here and download these cells in NeuroML format. Okay, and I'll just mention one more because if you are interested in doing modeling, this might be one of the ones that would be really exciting to you. If you're just getting started and you're trying to learn to use some new tools, if you're a student or something, then this might be a new cool thing to check out. So this project, NetPine, comes out of Bill Litton's lab, and Bill is he's at SUNY Downstate. He does a lot of really interesting modeling, but the cool thing about this particular resource is it's not so much a simulator, it's about making building models, running models, especially large-scale models, much easier. And it has support for NeuroML. Of course, that's why I'm talking about it. So let me just tell you a little bit more about it. So, for instance, you want to make a new model in NetPine. Or maybe you want to build on components from other models. Like say you want to use channels that somebody's already created for some other model, or morphology from somebody else's model. You can import those into NetPine. The thing that's most exciting about NetPine is then if you want to make a relatively complicated network model with realistic circuitry, different kinds of subcircuits represented, it's a tool for doing that. So it's a tool for basically saying I have these populations of cells and this is how I want to connect them, and it will do that for you. And then, you know, it lets you do this in 3 dimensions. So you can say where the cells are, how you want them connected, where on the dendrites you want them connected, and things like that. You know, it sounds like a simple thing, but it's actually a little tricky to write software to do that sort of thing. So you want to speed it up as much as you can. You want to use tools if they exist. And then what's really cool, whether you're doing a relatively simple model, or maybe you're doing a single cell model, but it's going to be really complicated. It's going to be very biophysically realistic. You want to have a lot of the morphology represented. You want to have a lot of different channels. You might want to run the model in parallel on some sort of supercomputer. Or maybe you have a hugely large-scale model where the cells are simple, but you have a lot of cells. Or maybe both. You know, your cells are complicated, but you have a lot of them, like the blue brain model that I showed you. NetPine will do an automated parallelization of your model for you, and it will output neuron code for running this simulation, you know, sort of automatically. So you can create this model, you can use pieces of other people's models or create them from scratch. You can then output the simulation code that you need to run these parallel simulations in Neuron. And one of the nice things about Neuron is it's well benchmarked. People have been using it for a long time. The computational schemes have been tested very well. So you can feel confident using it. And then it will bring the results back into NetPine, and you can do some visualization and analysis there, or you can use other tools to do that I'm going to show you in a minute. And then not only does it import NeuroML, but it exports NeuroML. And the important part about that is then if someone else wants to take pieces of your model, reuse your channels, reuse your cells in a different kind of network, they can do that. So it's very modular, that's the important thing. And it makes modeling much quicker. If you can swap in and out cells, like your cell models, you can test different things. You can swap in cells that are maybe pathological. You can change your connectivities very quickly in a way that you think it represents something that's pathological. These are ways that you can speed up modeling. All right, so it's important, if you're going to use a tool like this, it's important to be able to get models that are already in NeuroML format. We have a database for that. You can search there. There are a couple of ways of getting results. It uses keywords, but it also uses an ontology. So for instance, if I search on Purkinje cell, cerebellum, it knows that Purkinje cell is in the cerebellum. Maybe I want to see other models from, other cell types from the cerebellum. So it will return, other cell types from the same biological structure. And then once you click on one of those models to look at it, it gives all kinds of information about the model, including it will link out to ModelDB if that model is already in ModelDB. So you can get the NeuroML version here. You can go to ModelDB and get the original code if you want. You can run both, compare them, whatever it is that you need to do. In this case, this particular model is on ModelDB. It's also at Open Source Brain. And so if you click on these, it'll take you to those platforms so that you can look at the model there. It will also link to PubMed for the publication, so you can learn more about the publication. You can get the files. For many of these models, we also have figures here that show what the model behavior is under different kinds of protocols and things like that. And we have thousands of models here. So, I think it's a pretty good resource. And this is just to show you, like, if you link to ModelDB, what would you see? Well, you would see the ModelDB page for this model, the original code, and then vice versa, ModelDB, if a model in ModelDB is in the NeuroML database, they also link back to our files. So you can click here and go to our database and get the NeuroML versions. And we're trying to really ramp up automated conversion of a lot of models so that more of the models that are there will be in NeuroML. The only reason for that is just so you can use all the tools that support NeuroML. Okay, and then just to quickly mention that blue brain model that I showed you with all those different morphology types and all those different morphology, electrophysiology types, 207 different types of cells, all of those are in this database. So you can get all those blue brain models and run them. Okay, so I want to tell you a little bit about open source brain. I just mentioned it briefly. If you're not, a computational neuroscience person, but you're sort of interested in modeling, you kind of want to check it out and see what might be interesting about it, this is a good resource. This is a good place to go. This is not my project. This is a project of Angus Silver. He is a professor at University College London. He's a neurophysiologist. But I'm sort of a collaborator. I'm an early user, and it's based on NeuroML, and some of our tools are built into open source brain. But it's really his project. I just want to make sure I give him the credit. So if you go to open source brain, a good place to start is on this front page. All of these all these projects that are listed here have tutorials with them. So you can click on any of these and you can go and you can run a tutorial and learn more about open source brain. Clearly, what open source brain is meant to do is be sort of a collaborative platform for doing modeling. So there are a lot of existing published models that are there. They're there as projects. There are wikis, so you can see things like, are there bugs in this published model? It'll say, yes, there are bugs, and here's what you can do to take care of them, or here's updated code without the bugs. So it's a good place to go and look for models if you think you're going to want to run it. Okay, so how many of you have ever used GitHub or some kind of version control system? That's good, especially a mostly biology audience, maybe. Okay, so open source brain is built on GitHub. GitHub is a social, they call it a social coding, platform. Basically what it is it's an online version control system where you can have an account there. It's free, and everything you put there is public unless you pay extra for a private account. But essentially what it is it's a nice way to share things with the whole community, but also to promote transparency, to promote reproducibility. So the science community has really, picked up on GitHub, and all kinds of labs, especially labs of younger people, are sharing all of their code through GitHub. And so the idea is, the way the version control systems work, if you aren't familiar with it, is you put your code there. Let's say that you want to change something. Well, you change it, but the code that you had previously is sort of still there. You can still backtrack to it. And so it's a nice way of sort of keeping track of all the things that you've done. If it's public, then other people can fork your project, which basically means they sort of can work on the same project, but along a different path. So they can do different things with what you already have there, but you can see it. If you're working collaboratively, then you can say, okay, I'm going to merge that back into my project. Even if you're not a computer science-oriented person and you don't want to use any of the versioning, it's a great place to share your code, right? So it's still a good place. What I like about it, secretly, I'll tell the PIs here, is that every student in my lab has to use GitHub, and so I can see what my students are doing, and I can comment on what they're doing very easily through issues, this kind of idea. So it's a nice tool to use in your lab that promotes open science. All right, so here's just a screenshot of one of these tutorials from a single cell model. And so just to show you, once you go into open source brain and you say, I want to simulate this model, this is the kind of thing that you'll see. This is built on Geppetto, which is a great resource that's very, very flexible, but it allows for pulling together a bunch of different software tools. Like here you can visualize your model while you're maybe building it, and then you can go and run it with Neuron or something like that, and then you can bring your results back and show them. And the nice thing is that their visualization tools are really beautiful, and so you can make these fantastic figures for your papers and your talks. But so the tutorial walks through some of the things that they have here. You can do things like, I think this is visualizing distributions of channels on this reconstructed cell. So there are models of channels. Those channels are distributed, and you can see how the distributions work. You can run the model, and you can visualize results. They have the same kind of thing for networks. You can visualize your network. You can focus in on particular cells. You can look at the distribution of channels on those cells. You can look at connectivity. They have things like these connectivity matrices that you can use to see exactly how all the populations of cells are connected in your model. So it's just a nice tool for using in the classroom as well. All right, so one of the things that I wanted to be sure to mention is that, I was talking earlier about NetPine, and so one of the things that you can do with NetPine through Open Source Brain is you can, create a model in NetPine, bring that NeuroML model into Open Source Brain. You can then automatically send that model to run in parallel at the Neuroscience Gateway. And so this is just a slide, of the website that you see at Neuroscience Gateway. But what Neuroscience Gateway basically is it's funded by government agencies, and it's a resource for the community. If you want to run neuroscience models, a lot of the code that you would want to use to simulate those models, like Neuron and Python and all these things, are available there. And so you can go there and you can simulate your models on these hugely parallel computers and then get your results back later. And so one of the things you can do is you can create a large-scale model in NetPine. You can then go have it as a project at Open Source Brain. You can automatically send it, because Open Source Brain, you don't have to have a special account or anything. Open Source Brain has an account with Neuroscience Gateway. You can simulate it on the gateway, get the results back, and do a lot of visualization at Open Source Brain. So it's a good way, it's a good entry point into large-scale modeling if you want to do something like that. Okay, so I want to return to the idea of transparency. I alluded to this in my title. I said testing the data-driven model. So there's a whole other aspect to, I think, computational modeling and data analysis and everything else that we might talk about here, which is we as a community probably don't do a good enough job of what in the software community would be called verification and validation. Right? So there's a whole area of computer science around this idea of making sure that when you're doing modeling, your code does what you think it's doing. And so I want to talk about another project out of my group. It's done in collaboration with my collaborator at ASU, Richard Gherkin, and I want to talk a little bit about that project just for a few minutes. Okay, so suppose I decide I want to, reuse a model of a particular type of cell, and I go to ModelDB and I say, oh, well, I'll just download a model from ModelDB and run it. I go to ModelDB and there are like 108 models there for this cell type. And I'm, oh, what do I do? Which one do I use? Which one does the things that I am interested in? Right? And so as an example today, I'm just going to, do something really simple, but I guess the point is you decide. You decide what is the model supposed to do? What are the things that I'm interested in? Some people are like, well, I just want it to be as biophysically realistic as possible. I want to just put all these biological details in there. Some people are like, well, I want a model that maybe isn't too complex, so I just want it to have the characterizations that are important for the questions that I'm asking. But the point is, you have to decide what are the important things it should do. I want to help you with the infrastructure for doing that testing, for deciding, does this model do these things that I want it to do? And so the conceptual idea here comes from the software community, the idea of unit testing. So if you're doing collaborative creation of software, like in an industry environment, unit testing is one of the things that you have to do all the time. So you create these little tiny tests, and you run it on your software, And then the idea is when I add some functionality to my software, I then run it through these unit tests again and make sure nothing is broken, right? That's the idea. That's how you make sure that your software is doing all these little things that it's supposed to be doing. And so then the idea in modeling is, or data analysis or anything else, what are these tests? And then I can, once I define the tests, I decide on the things that are important that my software should be doing, my model should be doing, then I can test a bunch of models against whatever that is. I can run these tests on a whole bunch of models. And so we're developing an automated framework for doing that. And NeuroML helps with that, right? Because we can, once we define tests, we can run them on thousands of NeuroML models very, very simply in an automated way because we know what all those models are supposed to be doing, right? It's because it's a standardized way of describing the models. But our framework isn't just for NeuroML. You can use it on anything as long as you have the code for multiple models. All right, so the framework is called SciUnit. SciUnit is actually domain agnostic. It doesn't have to be neuroscience, it can be anything. So it works on any kinds of models that you would be interested in or any kind of code. But I'm going to be talking about a subset of that called NeuronUnit, where we have a large number of tests designed specifically for neuroscience. So of course the kind of tests that you want to run against a neuroscience model are the same sort of protocols that you would do in an electrophysiology experiment. Because if you're making a model and you're constraining it against data that you obtain during an experiment, that's exactly how you want to test your model. But you could come up with any kind of test you want. All right, so I'm going to show you a really simple example where we took 8 models that are basically in the neuron code already. These were models for mitral cells. We obtained them from ModelDB. So these models were created by different labs to do different things. They weren't necessarily all created to be, biophysically realistic. So, that's sort of a caveat here. They weren't necessarily built to do what I'm testing them for, but let's say I wanted one that matched certain electrophysiology data and I want to know which one does the best. All right, so that's the idea here. These are all deterministic models. They don't have any noise or stochasticity. They all have spikes. They all create action potentials if I give them enough current or enough synaptic inputs. Some of them are quite simple spatially in terms of their morphology. Some are more complex. And then the idea was our tests are basically simulation protocols. They're sort of doing electrophysiology experiments in the computer, right, in simulations, so that we can compare to electrophysiology data. And in this case, we just chose 6 very, very simple things. We chose 3 sort of membrane parameters and 3 parameters that you might get from looking at an action potential. And we said, well, let's just say that we want the best model that fits these 6 parameters as an example. All right, so in order to run these kind of tests, you need data. NeuronUnit will actually use data from your lab, so you can use whatever data you want. But we have a lot of APIs. Those are sort of automated hooks for getting data from NeuroElectro, which I'm showing you here, but also for the Allen Institute database. So you can go there and you can get data from their database sort of automatically through our tools. In this case, NeuroElectro, if you guys don't know about this resource, it's really fantastic. So NeuroElectro, basically what they do is they do data mining of the literature. And so it's a cell types oriented database. So if you go there, you can look for different cell types from different regions. And what they have done is they have collated information from thousands of articles where they looked for these kinds of numbers for specific values in the literature. So as an example here, with the mitral cell, let's say we wanted to look at input resistance. If you click on one of these blue dots, what you get is something like this, which tells you what the value is, what the cell type is, what the paper is, where's the article that this data point came from. All right? And then then these values are across all the values that they obtained from the literature. So there's going to be a lot of variability. But you can dig into the literature and figure out, if you're happy with these numbers or not. But anyway, this, for the example that I'm showing you, this is where we obtain the data that we're comparing to with these tests. Okay, so how did these cell models do? How did these 8 mitral cell models do? Well, they're ranked here in terms of how well they matched these experimental values that we got from the literature. And what you're basically seeing here is each of these blue dots is one of those tests, and this is 2 standard deviations away from the mean that you might expect to see. So again, you have to interpret this in terms of the questions that you're asking, but the point is that this is a way of basically having a nice infrastructure for deciding what kind of tests you're going to do to constrain your model or evaluate models, and then you can share those tests so that people are sure they understand exactly what were the data analysis protocols that you were using. Yeah, so Simon O'Connor, this is his model. He's a really good friend of mine. So every time I show this figure, I'm like, ooh, maybe I should just cut that one out. But I think he just did not do a good job with this parameter. It's not very biophysically realistic. I don't know why he used a value that was so far away from experimentally recorded values. Maybe he chose some of these values from the literature and then he fit his model to some data, and that just was the best fit. I mean, a lot of times that's what we do, right? But so that's...
He doesn't understand yet.
Huh?
Nobody understands yet.
Well, I would just say, if you were looking for a biophysically realistic model, you probably shouldn't use this one. In contrast, these are models from Gordon Shepard's lab, done by Michele Migliore, and he has a very nice large-scale model of olfactory bulb, and all of his cells kind of look like this. So he's done a very good job of constraining his models against the literature, and they're good models to use if you want a relatively complex cell model. You don't mind that it's going to take some time to run it, and take it how you want, but that's my interpretation of this graph, right? Okay. So you can find out more about Neuron Unit by going to our website. It's a work in progress. We're working on a lot nicer interface, which will probably be launched in about 6 months, maybe a year. Where you can go to our website and run stuff server side, run tests there. But for now, if you're kind of a hacker type, you can get all our code. You can run our tests. You can use existing tests, that sort of thing. So maybe something to keep an eye on in the future. All right, so I won't belabor this, but I like to kind of take a minute to and I didn't talk that much about reproducibility today. I talked a little bit about tools, maybe some interesting things for students who might be interested in starting to do some modeling, just letting you know what's out there. But I like to point out that there are things that all of us can do to help with reproducibility that we should sort of keep in mind, no matter what our area is, whether we're a data scientist or a computational modeler or whatever. And so one of the things is, if you're creating software, whether it's for others to use, or maybe just for a large group of people to use in your lab, you should always have something built into the software that keeps track of what you've done. So some sort of diary, some sort of way of saying, I did this manipulation to my data, and then I did this, and then I did this, and then I did this. We need to have good tracking of what we're doing. And the more you build that into software, the better. Another thing to keep in mind is, of course, if you're working in a domain where there are these description languages for complex models, so for instance NeuroML, or in systems biology there's SBML, which is a similar effort, right? You should collaborate with those people. You should use standards wherever you can. If you're just doing data analysis software, of course, make sure that everything you're doing goes along with this Neuroscience Without Borders new standards for electrophysiology. And then, of course, I said a lot about GitHub. Whether it's GitHub or something else, you shouldn't just be making your software available on your website. That should never be the case, and there are good reasons for that. And that is, as we move along in life and we move to a different place, that software might get lost, or we might think, well, I'm not that interested in that software anymore. I'm not going to maintain that website. If you have a publication out there about that software, for sure, that software should be available forever, right? So you need to put it somewhere where it's going to last, and GitHub or something like that should give you that. If you're a researcher or a modeler, I've talked about a lot of different things, but there are a lot of things that I didn't really touch on, things like using best practices in your articles. So I mentioned tables, automatically creating tables. As much as possible, if you have any numeric values, you should make them available in tables in your paper. And the reason for that is that then those values can be easily mined by automated software and made available in sort of some of these conglomerate websites that are gathering data from articles and making them available in a more usable way. You guys know there are so many publications out these days, right? I mean, how can you keep track of the publications in your area if there are resources out there that are going to get those important data points for you and collate them and make them available in a nice way where you can look across multiple journals and publications? That's going to be helpful to you. So some of these things I talked a lot about, but I'll just say that I think one of the other things we can do is we can keep track of people who are downloading our models, downloading the software that we make. That should be part of our productivity, right? This should be part of how we're reviewed as PIs, and so that's a good thing to keep in mind. And then the last thing is, as reviewers and editors, of course, one of the best things we can do for our community is to be careful about requiring code whenever we can, to try to choose journals, support journals that are going to support these kinds of efforts. And I think that, I've seen such a huge difference over the last 10 years. You're reviewing grants for the NIH, and you have sort of resource sharing documents that you have to look at. Some people are more concerned with them than others. As a reviewer, I'm that reviewer who's going to say, this was not complete enough. It's not good enough to release it on your website. You need to use all these community resources that are out there. Make your data available through those community resources. Okay, so I will stop there. Thank you so much. Thank you.
Please accept this small gift of appreciation from our college.
Thank you so much.
We have time for a few questions.
That's okay.
So I wanted to ask a question.
Yeah.
So you have all these models now collected. Are there any implications for big computing resources that are needed? Can we use the same big computing resources that already exist, or?
Yeah, so there are a lot of interesting answers to that question. So I think a lot of universities have very good computational resources now, good enough for maybe not the blue brain model, but a lot of the large-scale models that people would want to do. You can do them, on a cluster within your unit or a larger supercomputer at your university. Neuroscience Gateway is actually free, so people can use Neuroscience Gateway. I don't know if it'll last forever. If it doesn't last forever, there may be some issues around that. But also, there are a lot of people working on some really interesting neuromorphic hardware. So this is hardware that has been designed specifically to run these kinds of models very quickly and very well. And the parallelization is sort of built into the hardware. And those are some very interesting efforts. And actually, you can get a collaboratory at the Human Brain Project in Europe, even though we're in the US, we can get a collaboratory there. And you can use their neuromorphic platforms to run these large-scale models. And that's what some of the people in my lab are doing. So that's also an interesting approach. All right, thank you. Oh, one more. Yeah. There are certainly still people in the community who do not want to share their data or share their software. And of course, you guys can think of many, many reasons that they might give for doing that. There's been a lot of actual research on this that shows that people who share actually do better. They get cited more. They progress better in their career. There are probably a few examples in the more, sort of senior researchers where they already got to that point before we all expected everything to be open source. But in terms of junior faculty, I think they embrace this open source. I mean, you see it everywhere you go, this open source movement, and I see that they do well because of that. So I think really that is what is driving the change now, is just that there, in so many circles, it's really, there's a lot of peer pressure. There's a lot of peer pressure to be open with your models and your data, and people are seeing that they do well if they do that.
What you think is the best neurocomputational software for coupling with external mechanisms like magnetic fields, electromagnetic stimulation, ultrasound?
Yeah, so that's a tricky one. So if you're interested in local field potentials, there are some very nice tools out there for dealing with that. I'm not as familiar with stimulation, but in terms of recording, there's this tool LFPi, which does a very good job, and then there is another tool that interacts with Neuron where you can run, well actually LFPi uses Neuron too, but there are some other tools where you can take these large scale models and you can generate things like local field potentials. Like I said, I'm not as sure about stimulation. The nice thing about Neuron is it actually has, a coding language. So you can add things in. I mean, you could build in something that acted like the stimulation that, that you wanted in your model that was some sort of more magnetic or whatever. I'm not aware of anybody who's doing that right now, so that would be a very interesting thing. Yeah. Yeah. But reviewers. I mean, that's why it's not the best system. It's just like if you're publishing your clinical work, right? Who makes sure? Whoever is trusting you that you obtain the data the way you said you did and allows you to publish it because they think it's quality. It's the same. It's the same in computation. It's our community polices itself. When we do reviewing, we try to determine whether we think somebody did a good job creating and constraining and testing and validating their model. And if it's sort of a joint, maybe clinical modeling study, then maybe it's going to be more careful than if it's a basic science model, but I'm not sure. Yeah. Yes. Yeah, so the question is whether this sort of approach, this idea of reproducibility and rigor has reached other domains. To some extent, I think it's more prevalent in neuroscience and systems biology, more sort of biomedical oriented modeling is where you see it more. The ecology community does have some movement towards some standardization and putting tools together. I don't know as much about, outside of biology. Yeah. Actually, there are, I know like in astrophysics, they have I've seen some of the things that they're doing around reproducibility. Yeah.
Are you aware of the virtual population by the FDA and Zurich MedTech, which is a complete computational model of the human brain?
No, I don't know anything about that model. I'd be interested to look at it. It must be maybe more cognitive or at the...
No, it's anatomical and neuroanatomical.
Okay. I would love to look at that, so maybe you can fill me in, or I can look it up. Thank you. Yeah. Yeah, so, well, one of the things that we're doing is, for instance, at the NeuroML database, right, it's really easy for us to run a billion tests on, different models that we have. We can just run whatever we want, and we can decide which of those we might want to share with the community to make it easier for them to identify models that they're interested in. If you are developing new models, one of the things you might be the most interested in is using these unit tests as a way of determining fitness of models when you're doing fitting. So we also have routines for fitting models, like doing genetic algorithms and some other things, some other approaches where you use the tests as the way of determining the fitness of the model. Yeah.
Thank
Okay,
you
I'm there.
again.
Thank you.